Well, hello everyone. I'm very happy. I'm pleased to share my research today. Today I want to share with you what we've learned by studying the viral and microbial communities in acute exacerbations. In particular, the case of Achromobacter colonizations in the CF lung. So as Dr. Rower introduced, with our cystic fibrosis rapid response, we studied acute exacerbations of around 10 patients. And one thing we noticed by looking at the microbial communities is that some of them were dominated by Achromobacter. So Achromobacter species are a problematic microbe in cystic fibrosis. Unfortunately, two of these exacerbations were fatal, and they were dominated by either Achromobacter roulanderi or Achromobacter psilosoxidans. We also observed two other exacerbations, one for patient CF032, which is for the patient that we're currently following with the hope to apply phage therapy. So yes, we know Achromobacter dominance in cystic fibrosis is a problem. Why is that? We don't know the exact answer, but here is a scanning electron microscopy image of Achromobacter growing. And we know that these bacteria can grow using different metabolisms. It can grow under aerobic and anaerobic conditions, which maybe help the microbe adapt to the CF lung. It is very good at obtaining nutrients, so it has mechanisms for nutrient scavenging, which means just getting rare nutrients from other sources. It has intrinsic and acquired multi-drug resistance, which means like several efflux pumps are present in the genome. And the thing that we think is why it's making the CF exacerbations so bad is that it encodes hemolysins, which are proteins that will directly attack the host tissue. So all these are general characteristics of Achromobacter, but what is unique about the Achromobacter strains that we identify in our CF exacerbations? So we went on to study exactly the mobile elements. So the mobile elements, as Dr. Rower introduced, are parts of DNA that can be moved around either by phages or plasmids or transposons or genomic islands. So all these pieces of DNA are moved around the environment and they can get integrated into the microbial genomes. So we studied the four exacerbations that I show you. We studied them by sequencing the total DNA metagenomes present in the sputum and also by sequencing the genomes of the clinical isolates of Achromobacter. So we were able to identify the specific parts of the genome that were different between the Achromobacters that were growing in the sputum and the Achromobacters that are present in the databases. So here on the x-axis, we see the genome of Achromobacter roulanderi, and this strain is in the reference databases. And all these black parts are the pieces of DNA that were present in the sputum of patient CF116. So as you can see, we have coverage across the whole microbial genome, but we do have some pieces of DNA missing, which means that these parts of the genome were excised or deleted in the CF microbe. So this shows us that we already knew, but we are showing in cystic fibrosis, like microbial genomes are very dynamic. So they are losing parts and they are also winning parts. So we see all these kinds of deletions in the four patients that we study. In the case of 116, the closest reference genome was Achromobacter roulanderi. And in the case of the other three patients, the closest reference genome, as you can see, was the same, for example, for these two patients, but the regions that are excised are different. So this is telling us about unique adaptations inside each one of the patient's microbes. And all these deletions, as we talk about, they look like phages. So they have either a lot of hypothetical proteins or phage-related proteins. Now, about the other dynamic part of the genome are the insertions. So insertions, we identify them by comparing against the reference genome to what we could assemble from our metagenomes from CF sputum or from the clinical isolates. So for example, here we see this genomic region that it's only present in the pieces of DNA that were isolated from CF sputum, but they were not present in the closest reference genome. And this proportion is not insignificant. It's around 4 to 6% of the total genomes. And this is important because they also look like phages, and what phages can be doing when they are integrated inside the bacterial genomes. So when a phage is residing inside a bacterial genome, it's called a prophage. And prophage can provide several mechanisms to help the fitness of the bacteria. So for example, here in this review paper, we see several of the mechanisms by which prophages can help the host, which is the Achromobacter. So they can confer superinfection immunity, which means that when the prophage is inside the genome, it prevents the infection by other bacteriophages. They can eliminate other bacterial competitors by secreting molecules to fight other microbes in the environment. They can do horizontal gene transfer, which is just sharing their genes with other microbes around. They can enhance the antibiotic resistance mechanisms, which means they can enhance the function of efflux pumps, etc. They can encode virulence factors, like the case we saw before with Shiga toxin induction that was encoded by a phage, or they can alter the gene expression of several genes in the bacteria. So this is why it is very important to characterize these specific insertions that look like phages, because they could be affecting any of these functions inside the Achromobacter genomes. We characterize those insertions in our 4 case studies, and we have around 12 to 20 new insertions in these genomes. And I say new because when we compare these insertions to whatever it's on the databases, we rarely see them as a whole. So we think that these insertions encode phage proteins, and they are new phages. So here we see 2 that look like very much like phages. For example, in Achromobacter, isolated from patient CF418, we see several insertions and only very few phage proteins annotated as phage. But what it means is that many of these hypotheticals could be phages that we don't know their characterization yet. We also see, in addition of phages, we see several conjugative elements as plasmids that could be, that are new insertions in the genomes. And I'm going to show just one example of the genome annotation of each one of these. So here, what we were talking about, the specific ways phages can modify the host metabolism. So for example, here in just this phage isolated from CF116, we see all the components to create a phage capsid, which is like a phage should have them. We see integrases, which are the proteins that the phage used to get inside the phage genome. But we also see some proteins that could be affecting the host. For example, this mannitol transporter system. So this mannitol transport system could be helping the bacteria to internalize the sugar mannitol. And we see several examples of these phages, of the potential of these phages to be manipulating the host genomes. So for example, in the phage insertion 10 from Achromobacter CF418, we see all these genes that are in red with R for iron scavenging. So they could help the bacteria to get more iron from the environment, which will help the bacteria grow better. And we see all those kinds of modifications inside these phages. And now you can see that here some names are in gray and some names are in black. The ones that are in black are where we really know the annotation. And the ones that are in gray is where we use the conserved domain database or other more sophisticated bioinformatic tools to really try to guess what each one of these functions are doing. And even we use all the best phage annotation tools, we still have a lot of black open reading frames here, which are the unknowns. So I'm showing you that we have inside the genomes of these Achromobacter strains, not only new phages, but are also a lot of new uncharacterized phage proteins. And we think that these prophages are active inside the CF lung during exacerbations, because when we count the number of viruses per each bacteria, in the stable cases, the virus to microbrevities are around 5, but in the exacerbation cases, these numbers go up. So it could mean two things. It could mean that there are more viruses coming from the outside, but the most likely explanation is that all these prophages that are inside the microbial genomes are getting induced, so getting expressed. And there are several mechanisms by which prophage induction can happen. They can happen by oxidative stress, but one of the mechanisms that this can be mediated is by antibiotics induction. So we did a test in the lab to see if we could induce these prophages inside the Achromobacter genomes by using antibiotics. So here you can see time on the x-axis, and then on the y-axis, the bacterial growth. So we obtain, we grow the clinical isolate of CF116, just happily growing by itself. You can see the growth curve is here. And there is some classical inducers that have been shown to induce the prophages, such as mitomycin C. So when we grow the bacteria, in the presence of mitomycin C, which see the bacteria growing, and then we see a decay. And this decay, it's because phage particles are induced and they blow apart the bacteria. So we see a phage induction event, which is with a known phage inducer. But we also tried that with the antibiotic colistin. So we grow the bacteria with colistin and we see a phage induction event. So this is just like one of the mechanisms in vitro about how these prophages could have been induced, but there are many, many other mechanisms that, that could induce prophages. So now talking about the prophages that are inside the bacterial genomes, we went on into the part of, well, can we use the phages to actually control the Achromobacter infection, and then the use of lytic phages as therapeutics has been proposed. So how we did it, it's with a great team of undergraduate students. So phages are everywhere. So we obtained the Achromobacter strains from the clinic. So this Achromobacter CF116 from the clinic. And then the phage hunters team collected several samples, mainly from water sources, for example, from this fish pond at San Diego State University, and then filtering the water to get rid of the bacteria and growing together. The water sample with the clinical isolate, here we see just the bacteria growing. And after several times of trying this with several samples, finally you see this clearance area, which means that here are all the bacteria growing, but here the phages are killing the bacteria. So yay, we have a phage. So now the phages, we know we have a phage that can kill this bacteria, and we start the characterization of the phages. So for example, here, this phage that was isolated from the fish pond is Achromobacter phage kiwak. This is how the phage looked like by electron microscopy. But as we mentioned, it is not enough to just know that this phage can kill the bacteria. We want to know what's inside the genome. So we set up to sequence each one of these bacteriophage genomes, and we did it with two approaches of sequencing. One is Illumina sequencing, and the other one is this very small sequencer, which is called the nanopore sequencer. And the advantage of doing this sequencing is that it is fast. So in around 2 or 3 hours of starting the sequencing, we can get the genomes of the bacteriophages. So here we have Tram Lee, who isolated several bacteriophages, putting the phage DNA into the sequencer, and then sometime later we get the genomes. So now we in our case, so this was some time ago, we combined the nanopore sequencing with the Illumina sequencing to get complete phage genomes assembled. So here are all the bacteriophage that we obtained, and we named them after, they are called the Kumeyaay collection. So the Kumeyaay collection, it's named after the native people of San Diego, which are the Kumeyaay, and we used several of the Kumeyaay words to just name the phages. So we have the complete genomes. Mainly, the main reason we wanted to get the genomes is to know, are there any toxins? Are there any integrases? Will they become prophages and so on? So besides doing that analysis, we also compare each one of these Achromobacter phage genomes to other phages in the databases. So here we have a phylogenetic tree, which is telling us about the relationships between each phage. So here with pink dots, we have the bacteriophages that we isolated in our study. But we also, by doing this bioinformatics search, discovered there were other Achromobacter phages in the databases, which is great. So we have Achromobacter phages in the Kumeyaay collection from 2 clusters. And this is important because we want to have like a large number of phages in case the bacteria becomes resistant to each particular phage. So now it's an example of great scientific collaboration. All these other phages were reported by the group of Dr. Christine Poussel at the Université de Paris-Sud, and she was very kind to share the phages with us. So now we have like a larger collection of bacteriophages that we can screen to see if they kill each one of the Achromobacter strains. So in going more into detail about the genome comparisons, we see that these 2 Achromobacter phages that we have, they look alike, but we also have some variation, right? So this protein, for example, has variation. These proteins, some of them are present, some of them are not. So it is relevant to know all this variation and the relationship between other phages, because we could compare these variations later with the host range of about which phage infects which bacteria. So that's what we did. So we tested the infectivity of our phages and the Côte d'Ivoire phages, which are the ones that were sent to us from France, against 25 Achromobacter strains from the San Diego CF clinic. And with this big collection, we get around 92% of the strains are lysed by at least one of these phages. So it is great, and the whole purpose of doing this is because, as I mentioned, we had like 2 fatal exacerbations. So we say, and in that case, we were slow to harvest the phages and get them. So in this case, we say, okay, we have a collection. If a patient needs them, we could eventually use them. So that is the case, and we are following patient CF032. So we've been following this patient for around, well, we have data for this patient for around 4 years. This patient has been colonized by Achromobacter since 4 years ago, and we have 2 isolates from that time. And more recently, the patient has still Achromobacter colonization. So now all this data comes from the clinical lab, so it's what the clinical lab can grow. But our main question was like, is that reflecting what's going on in the lungs? So we obtained 2 sputum samples and obtained total DNA metagenomes. And we corroborate the hypothesis that yes, Achromobacter is a dominant microbe in the lungs of this patient with 93% relative abundance and 88% relative abundance. So we obtained the metagenomes, we obtained the strains from the clinic, and the clinic reported that these 2 strains are resistant to antibiotics. So here we have the antibiotic resistance for the first isolate we have, and for the second isolate, which is only a couple of months after this isolate, we see like the resistance to the antibiotics is going up. So this is one of the cases where we want to use phages to kill Achromobacter in the lungs of the patient. So we tested, we obtained the 4 strains from the clinic. These go from older to newest. And then we tested the infectivity of the phages against each one of the strains. We tested our Kumeyaay collection, the Cote d'Ivoire collection, and another collection, which is called San Diego collection, for which we have 5 phage genomes. So we can see that we have at least 1 phage that infect each one of these 4 clinical strains. And this is how the tests look like. So here we have a plate where we have Achromobacter AC5204 growing, and then we put a drop of each one of the phages. And if we see clearance, it means that the phage it's capable of killing this microbe. And we are doing all this work in collaboration with Dr. Anka Segal's lab, which she has a great team, which is developing further these phages. So we pick 4 phages from all these phages that can infect the strains from CF032. We pick 4 phages, trying to get them from different clades, with the idea that phages that are similar, so the bacteria can become resistant to the phage. So with the hypothesis that if they are similar, if you put similar phages, it will be easier for the bacteria to become resistant. So we pick 4 phages from different clades with trying to overcome this limitation. So we have several resistant mechanisms that can happen, and that's why we pick 4 different phages from 4 different clades. But basically, at each step of the phage infection cycle, you could develop resistance, you as a bacteria. So the bacteria can change the receptors, the other phages can chop the DNA, and so on. So that is when studying the genomes of the clinical isolates become important, because it could happen. So inside the bacterial cells, there are also war between phages. So for example, if a prophage, it's already integrated inside the bacterial genome, that could prevent the infection by a second phage, by mechanisms known as superinfection exclusion. So we have to think about all these aspects when developing phages that we want to use for therapeutics. So what we did is analyze in detail the genomes of each one of these 4 clinical isolates. So here we have a similar analysis that, as I showed before, we have on the x-axis a reference genome, the closest reference genome, and each one of these black regions, it's which parts of this reference genome are present in each one of the clinical isolates. So these were the oldest clinical isolates. These are the newest clinical isolates. And as you can see here, there are deletions that are different. For example, here we see that this large part of the genome is not present at all, whilst here some of them were present. So adaptations are happening not only among CF patients, but inside the lungs of one CF patient. Across time, we can see that the genomes are changing a lot. So here I'm showing you the deletions, and we can also see what is new in this genome. So we can see the insertions. So again, here we have a representation of the four genomes that we have, the history. And if you see white, it means that it's very different from the genome that it was aligned to. And sometimes the regions are not present at all. So again, we see that here, for example, in the most recent isolate, we have new regions that were not present in the previous isolates. And that's happening across the whole genome. So when we annotate these insertions and these deletions, again, we see a similar pattern. They have a lot of hypothetical proteins. They have some phage proteins. So they could be phages moving around across time in inside one of the patients. So we want to put all this information together with the infections patterns that I showed to see are these holes, are these insertions and deletions phages that are preventing the infection by the lytic phages that we want to use. So all this, it's really relevant to understand how phage therapy will work in the CF lung. So once we try to tear this apart, we selected those 4 phages that we say, well, we think that if we select them from 4 different clades, it will work. So we went on to the large scale production of phages. So from having, so all these experiments that I showed are in a small scale. We have, I don't know, 1 milliliter or 2 of phages. So now we want to produce a lot of phages. So we start with around 1 liter of phage. And then we concentrate all this phage lysate. We put the phage, the bacteria, let them grow. We concentrate them. Then we do a buffer exchange. And then it comes an important step that when you are producing phages, you are lysing the bacteria. So all the bacteria debris that it's there, you need to remove it. And in particular, one of the components, it's the lipopolysaccharide or endotoxin that you want to really remove from the phage prep because LPS can trigger the immune system. So we want to remove it. We remove with a 1 octanol, and then we remove the 1 octanol by dialysis, and we quantify the phage and the endotoxin units. We make sure that we have very, very low endotoxin units. And after we go through all these steps, we also sequence the clean genome lysate. Why? Because we want to make sure that during our phage productions, we didn't induce phages from the parental strain. So basically that we have in our phage lysate only the 1 phage that we want to apply and not other endogenous phages. And then we want to make sure that we don't have a mixed community. So that's from the research and production part. But after that, there is a lot of work to get these phages to the patient, which is all the FDA approval process, which Dr. Doc Conrad is going to talk about in his session. So all this work, all these protocols and everything were developed by Marisa Rojas in the Roar Lab, and we're doing all this with all the support from the Segal Lab at San Diego State University. So that's where we are at with trying to get these bacteriophages to the patients. So what we learned is that there is phage lytic behavior inside the CF exacerbations. The dominant microbes in these CF exacerbations are lysogens, which means that they are carrying several prophages that the prophages are new by themselves, but they are also carrying several uncharacterized toxins. And it is, we think it is relevant to further characterize all these mobile elements in the CF lung. And we see, unfortunately, that, well, Achromobacter is an emerging pathogen. We think it's causing direct tissue damage via hemolysins. And the prophage induction during the production of phages that we want to apply as therapeutics needs to be evaluated every time that we want to do a therapeutic application. So, of course, this is the work of many individuals. So I'd like to thank all of them. So the CF Rapid Response Working Group, Dr. Cynthia Silveira, Dr. Sean Bendler, Mark Little, Gina and Eugenia, which help us with all the ordering in the lab, we couldn't get done. Jenna Milk at the UCSD clinic. And then we have the Achromophages working group, which are the phage hunter, Tram, Marisa, Dr. Lily Han, Jessica, Jake, Lorena, Lance Bolling, which was able to isolate phages for 2 of the strains that were really hard to isolate for, and Dr. Helena Vilela. We got water samples from several places like wastewater treatment plants. So Alejandro Vega helped us get those to us. And when we need to do the sequencing really fast, Dr. Kristen Jepsen at the UCSD Institute for Genomic Medicine, it's always very helpful to get our sequencing done as fast as she can. And of course, Dr. Forrest Rower, Dr. Doc Conrad, and the lab of Dr. Anka Segal, Dr. Sherhao Hung, who did all the transmission electron microscopy, those beautiful pictures of phages. It's all his work, Dr. Greg Peters, and more recently, Nicole Jacobson, who's doing all the phage production work. The bioinformatics, we have support from Dr. Rob Edwards' lab, Dr. Liz Dinsdale, and Dr. Dwayne Roach. And our funding, our initial funding came from the Sprance Foundation. And then thanks so much to the Cystic Fibrosis Research Inc. My initial funding as a PhD student came from CONACYT and from UC Mexus. So I'll be happy to take any questions.